Students often say, this may be a stupid question, they start saying. And I say, no question is stupid. Only answers can be stupid. So students don't have to worry. It is us we have to worry when we try to answer. There's no question which is stupid. Every question has some value for which either there is an answer or there is no answer. So it's a great pleasure for me. This is the topic, as you know, and as you may have realized, is somewhat different from the kinds of things you do every day. But I suppose you invite me because you want a change. So I'm going to talk to you about something very different from what you are used to in your everyday scientific activity. I work on an insect, and this is a social insect. In fact, it's a wasp. Its common name is the Indian paper wasp. And I have spent more than 40 years trying to understand everything humanly possible about this insect with as little money and facilities as possible. This has been my goal, and I will elaborate on this a little bit. And I want to talk to you about it. Now, there are many ways in which I could discuss this insect society. But the way in which I want to discuss today is that these insects are very good at making a fine balance between conflict and cooperation. They have both. And that's why I have called this War and Peace, Conflict and Cooperation in a Tropical Insect Society. Let me begin with Yes. Let me begin with some very brief introduction. Most of you probably know this, but very briefly, what are insect societies? As many of you would know, some species of insects organize themselves into what we call societies. And I think it is legitimate to call them societies because they have many kinds of resemblance to our own society. They have the same kinds of problems that we have, the same kinds of solutions that we often have. So it is legitimate to call them insect societies. Perhaps the best known example of insect societies is the honeybee. Everybody knows about the honeybee. Honeybees live in large populous colonies with tens of thousands of individuals. This is a honeybee which I should say a little bit about because this is a typically Asian or Indian honeybee. There are basically four kinds of honeybees. One is was in Europe and now has been transported to US and Australia, but there are three Asian species, and this one is the most primitive of the honeybees. This is called Apis florea. It's also called the Asian dwarf bee. It's the smallest bee. It has very interesting features, and this is one species in which we could contribute a great deal of knowledge. But I think except one PhD student of mine, almost nobody has worked on this. Now there are a few people working, but there are very interesting features about these insect societies. In any honeybee colony, there are three kinds of bees. There is a single large fertile bee. She is the only one who actually reproduces. We call her the queen. She is not the queen in the sense she is not the administrative head, but she is the reproductive individual in the colony, the only reproductive individual in the colony. Then there are a small number of male bees, which are called drones. And in fact, they are famous for being lazy. So drones are usually very lazy. They don't do much work. They, of course, transmit their genes through sperm and they die. They do not participate either in domestic or in social life. And this is true of ant societies, bee societies, as well as wasp societies. In fact, I won't have time to go into this, but right in the beginning, I just want to say that even in the wasp that we study, males are lazy. So many years ago, one student came to my lab and she was very upset with the fact that males don't do any work and the females were to do all the work. And she said, I want to study this phenomenon. I said, sure. And she actually studied why the wasps are lazy. I won't have time to go into that thing because I'm going to talk about War and Peace today. But I must tell you that she not only discovered why the males are lazy, but she actually cured them of their laziness. So it is possible to study this. The rest of the colony consists of very large number of small, nearly sterile bees. These are the so-called workers. They do all the work. All the work that is required. They build the nest, they repair the nest, they clean the nest, they go bring food, they process the food, they feed the larvae, they defend the nest, They remove dead bees from the colony. Everything is done by these workers. They are small female bees. Now, they have in evolutionary time lost the ability to mate. So they cannot mate, but they have not lost their ovaries. In some other insect societies, they've also lost their ovaries. Here, they have not lost their ovaries, so they can still lay a few eggs. But in this group of ants, bees, and wasps, there's a very peculiar and interesting genetic system. They are both sexually reproducing as well as participating. So the queen bee will mate, gather sperm from the males, and store the sperm in her body in a little gland called the spermatheca. And she nurtures the sperm because the sperm have to be kept viable and active for years together. In fact, in some ants, we know that the sperm can be kept viable for 3 decades, which is something we are not able to do. And then whenever the queen wants to produce daughters, she uses the sperm. In fact, she opens the wall of the duct between the spermatheca, where the sperm is stored, and the oviduct, and some sperm will flow, fertilize the egg, diploid egg will be laid, resulting in a daughter. When she wants to produce sons, she will close that wall, no sperm flows, a haploid egg will be laid, it will grow into a viable reproducing male. So sex determination is through haplodiploidy. The interesting thing is the mother has complete control over the sex of her offspring, which none of us can hope to have. So this is what the colony consists of. And what you see here is in the center is the queen bee. She's clearly a bit larger than the rest. And you can see, surrounded by a group of workers. Now, if you watch a honeybee colony carefully, you'll find that the queen is always surrounded by a small number of workers. And we anthropomorphically say that these workers are on royal duty. Indeed, they are on royal duty because what they're doing is they're cleaning the queen, they're licking her, they're feeding her. She is too busy, she has no time to do any of these things because she is eating food, converting it into protein, converting it into yolk, converting it to eggs, and she can lay thousands of eggs per day. And she has to lay tens of thousands of eggs to keep this colony going. Now, if you mark these bees to distinguish one from the other, as we do with the wasps, as I will show you, you will find something very interesting. You will find that these worker bees which are on royal duty apparently seem to lose interest in the queen and go off and do something else a few minutes later, and other bees come and take their place. Now, this working in shifts has a very interesting consequence. The queen, in addition to laying eggs, the only other thing she does is she produces various chemicals which she releases outside her body, which are important for the regulation of the colony. Now, because these workers work in shifts, a very large fraction of the worker force has the opportunity to be very close to the queen, get these chemicals, go and distribute it to others. That is what happens with this. Now, here is a picture of a bee. Typically, honeybees work for the first half of their life indoors. They build the nest, they clean the nest, they repair, they process food, all of that. Now, the second half of their life, they go out in search of food. They collect nectar and pollen. Nectar is their only source of carbohydrate, and protein is their only pollen is their only source of protein. They bring it back to the hive, and in fact, they mix the nectar, they concentrate the nectar and make what we call honey. They add various antibiotics to it, and they mix honey and pollen and make what we call bee bread, which is fed to the larvae. The adult bees only take the nectar. And here is a bee which is transporting nectar and back. So in fact, these two structures are called pollen baskets. So bees do not consciously pick up pollen. As they go from flower to flower, pollen sticks on their body because bees have hairs to which attracts the pollen. And on the flight back home, they will brush all of this pollen into these two structures in their hind legs, which are called pollen baskets, and deliver them at home. One of the reasons why evolutionary biologists like me are fascinated with social insects in general, and honeybees in particular, is because of what is shown on this slide. When a honey, as you all know, honeybees sting. When a honeybee stings you, it's a little bit painful for you, but think of the bee. For bee, it's an act of suicide. When the bee inserts the sting into your body, as it is done for this volunteer here who posed for this, who allowed to be stung for this photograph, and when the sting goes inside, when the bee tries to withdraw the sting, it is unable to do so. Because the sting has barbs pointing outwards, the sting stuck inside, the abdomen of the bee ruptures, the bee flies away only to die within a few minutes. But the sting and the poison gland, which are still stuck on the victim's body, is still innervated. And the poison gland will continue to pump venom into the victim. And people have actually measured this for 60 seconds after the bee has flown away. So next time you are stung by a bee, don't chase that bee. That bee is not likely to do any harm either to you or to anybody else future. If you have the presence of mind, locate this sting and poison gland and remove it from your body before all the venom goes into your body. Now, this, of course, makes this an extremely efficient venom delivery mechanism, but at the cost of the life of the deliverer. And the question arises, how does natural selection perfect such an adaptation, which is not survival of the fittest, but survival of the one who is willing to die? And this is the paradox that attracts many of us to study social insects. So honeybees are not the only social insects. Ants are even more famous examples. There are between 4 and 15 species of honeybees, depending on which expert you ask. But there are 15,000 species of ants, and all of them are social. All of them have a queen, all of them have lazy drones, and all of them have this kind of organization. Now, there are some differences. One of the things that the ants have done, when I say done in evolutionary time, through evolution, is that different ants, different worker ants who do different jobs have different body size and shapes and structure. So there is polymorphism, which allows them to efficiently do certain jobs, although they cannot do other jobs. And all of these are actually members not only of the same species, but of the same colony. And you can see they're so different. For example, this ant would be very good at picking up small seeds and bringing them back to the colony, but would be hopeless as a soldier. Whereas this ant would be a very efficient soldier, which will actually guard these ants as they bring food back to the house. So division of labor, which is there in all insect societies, in the case of many ants, is accompanied by morphological variation, morphological adaptation suited for this. And sometimes this morphological polymorphism or dimorphism is taken to such an extreme. And in fact, the most extreme example is this Malaysian ant. A friend of mine, Mark Moffat, took the largest member of the colony and placed the smallest member of the colony on the head of the largest member of the colony before taking a scanning electron. It turns out that this individual is 500 times the weight of the smallest individual. They both belong to the same species. And I'm very fond of telling people in biotechnology and developmental biology that these two individuals have been produced from the same genome. The difference is entirely epigenetic. And how this happens is something that actually only now beginning to be investigated. Then we have the third group, which is my favorite group, the social wasps. I told you about bees, I told you about ants, and now I want to tell you about wasps. Most wasps are not social. Some of them are social, and the social wasps can legitimately be called paper wasps. And the reason why they can be called paper wasps is because they make their nest not from wax as honeybees do, not from leaves as ants do, not from soil as termites might do, but in fact from paper. This is a paper nest. Now, where do they get paper from? They manufacture paper. How do they manufacture paper? Much like how we manufacture paper. They scrape cellulose fibers from plants, chew it up, add various secretions, make it into a fine pulp, spread it into a thin layer and dry. And it is indeed paper you can actually write. What you see here is a picture of one such paper wasp. This is actually a colony that was found on the campus of the Indian Institute of Science, which I collected. And the whole colony is covered with a paper envelope, leaving only a small opening for the wasp to fly in and out. But if you open this paper envelope, you will see what I call a multi-storied apartment complex. There are several homes, first floor, second floor, fourth, fifth, sixth. All of these are made of paper. And these are used for them to raise their brood. What you see here are the pupae. Now, the difference between wasps and bees is that bees do not store honey because they are carnivores. They eat other insects and they have not invented a refrigerator. So they do not store food, which also means that every day the workers will have to go and successfully find and bring enough food to feed the thousands of individuals. Honeybees can store 2 kilograms of honey and say we can enjoy the winter. But these wasps have constantly they have to work. This is the wasp that I work with. This would be, I've collected some of these, but they would be extremely hard to work. First, they're very, very aggressive. Second is they are very shy. They cover their whole mesh with an envelope and you can't see anything. The nice thing about these wasps which I work, which is called Rhocoledia marginata, is they are open. They do not have an envelope. They're also paper wasps and they are small in number. So at best, you would have 100 individuals. Typically, you would have 20, 30 individuals. And therefore, they're very convenient to study. And because they do not cover themselves, you can observe all the adults and the brood and the interaction. So, for example, you can see here, these of course are the adults. These are larvae, there are pupae, and some of the cells have eggs. So all stages of brood, and you can actually study. Many people ask me, why should you study insect societies? And I think it's important to answer these questions rather than to say, of course it's important. Especially when public asks you, it's important to answer these questions. Now, there are many reasons to study insect societies. There are many, many applications that arise from studying insect societies in pharmacology, in physiology and medicine, in telecommunication and division of labor, in decentralized work organization, in robotics and artificial intelligence. I won't have time to go into this, but wisdom, knowledge from the study of insect societies have actually been applied in many of these areas, and millions of dollars of worth of money can be made from applying this. In fact, in computer science, there is a whole branch called ant-based algorithms. So algorithms based on the behavior of ants and bees are being used for efficient programming. But in addition to applications, I am much more of a basic scientist. So there are many more things for sheer curiosity. Ask yourself, how can we possibly turn away from little insects which are either fighting for a territory, sacrificing themselves to save each other's lives, practicing agriculture, caring for the injured, building structures with exquisite architecture, and so on. The list goes on. So there are many, many interesting things they do. And curiosity is the driving factor. For me, it is not the applications, but it's the curiosity that drives my interest in studying social insects. People also ask me, but how do you study them? Now, it depends, of course, a little bit on the species you're working with. So I will answer with reference to my own species. First and foremost, we can mark every individual wasp with spots of colored paint. And this is a very systematic way in which we use a fixed number of colors, a fixed part of the body, and therefore every wasp has a name. And there are computer files in my system for thousands and thousands of wasps, where we have the full lifetime record of what each wasp is. So each wasp has a name, so you can mark them in this fashion. And once you mark them, you see a whole different world, because wasps have individual personalities. These wasps are convenient in many other ways. One reason why they're so convenient is they do not live far away in the forest. They like to live around human habitation. In fact, many of them come and build their nest voluntarily in my lab, in my office, in my home. So you can study them wherever they are, but it's also equally easy to bring them to the lab and put them in different kinds of cages, including the plastic boxes, and they'll seem to live happily there. So you can do experiments of different kinds. I'll show you examples of some of these experiments. And you can stack many, many cages. This is my lab. I have called my lab a Vespiarium, place where we grow wasps. The unique thing about this lab is it has no walls. The entire room is covered with wire mesh. And the wire mesh is very special. The wire mesh has been chosen so that the size of the mesh is such that my wasp can freely fly in and out, but a bigger wasp, which the predator, cannot come in. And that predator is none other than that wasp with the closed nest that I showed you earlier. That was called Vespa tropica, which is also very common. And that wasp eats my wasp. So I have to keep that wasp away. And so I built this kind of a. In fact, it turns out that after some time, these cages became too small for some experiments. And I will show you one such experiment. So we built these large walk-in cages. And walk-in cages are very convenient because I can not only put a wasp colony in it, but I can also put the student inside. So the observation can go on. In fact, you can see my student sitting there and making observations. Both are inside the cage. Now, a little bit of how we study them. A little bit more about how we study them. As I already said, I'm going to focus today's talk on the balance between conflict and cooperation. That's going to be the sort of theme of my talk. But even before I get into that, I want to say a little bit about the process of doing science in general. I believe that the process by which we do science is as important as the product that we produce from the science. In fact, I would argue that the process is more important than the product. Unfortunately, scientists, when they speak either to each other or to the public, do not emphasize the process. They are only emphasizing the product. And this, I think, is unfortunate because if you don't tell the process, only the product, science sounds like magic. And this creates a barrier, not only between scientists of different disciplines, but also between the science and the public. And therefore, the process is very, very important. Secondly, the product of your research may be of interest to a small group of people, but the process is likely to be of interest to a wide area. So it's very important to emphasize the process of science. And that's what I'm going to try and do in this talk. So, in fact, every time I give a talk, I make the process completely transparent. I want actually all of you to participate in the process. And in fact, I'll show you how you can participate in the science that we do in my research. How do we do research? So there is this wasp, we mark the individuals, and it's absolutely fascinating. Because as I said, there is cooperation, there is conflict, there is work, there is fighting, there is disagreement. It's great fun to watch. So basically, we watch the wasp for fun. And you can spend hours and hours doing that, you will not. In fact, the interns who come to my lab are absolutely fascinated because for the first time they're sitting and watching. In fact, initially when the students come, I said, don't have any question. Don't read any books. In fact, my PhD students are forbidden from reading my books. Because you read my book, then you continue what I have already done. I want you to think for yourself. So watch these wasps for fun. It's a great entertainment. But as you keep watching, some questions will arise in your mind. And say, why did that wasp do something like that? Or how did this wasp manage to do this? Now, many of these questions we realize immediately are hard to answer. They're not easy to answer. But every now and then, a question arises which we think we might be able to answer if we did this experiment or if we did that kind of observation. Then you follow that lead. And these questions are different for different students, different for different people. And then you try to design a more structured, a more formal study to try and answer the question. Often we fail in spite of that. Sometimes we think we have an answer. I always say we think we have an answer. Science is always work in progress. Never be confident completely of the answer that you have. And invariably, if we even have a potential answer to a question, a tentative answer to a question, several new questions arise. Questions which didn't exist earlier, questions which you put in a thought of earlier, because you didn't know this answer. Now, you will feel that unless I answer those questions, I cannot really have confidence in the answer to the first question. So you pick the next question and you try to answer that question. You have an answer to that question, but you can say, but how can that be? Because I don't know how that works. So this is how science proceeds as a set of questions and answers. And there's no reason why I should not organize this talk as a set of questions. So I will give you kind of real life examples of what questions arose in our minds, what kind of study we designed to try and answer this question, what kind of tentative answer we had, and why we did not have confidence in those answers. What were the questions which arose in our mind because of which we were not sure of our earlier answer, and how we proceed. So I want all of you to participate in this. In fact, I want you to think, would I have asked a different question? Would I have done a different experiment? Would I have come to a different conclusion from the result of this experiment? Because there is nothing mysterious. This is something that everybody can do. So I want you to participate in this. I have said war and peace, so let me begin with stories of peace, and then I'll come to the conflict towards the end of the talk. Another nice thing about this species, which is not true of honeybees, which is not true of most ants and most other wasps, is that in this kind of species, the honeybees and the ants are called advanced insects or highly social species. The kind of wasps that I study are called primitively social insects, primitively used social insects. The difference is that in the wasp I study, all individuals look identical. The queen looks like everybody else. In fact, everybody is equal. And through social behavioral interactions, one individual temporarily becomes a queen. She's the only one to reproduce. Everybody else works for her. After some time, there is another individual. So this means that interactions happen at the adult stage, which decides who will become a queen, who will become a queen. Unlike in honeybees, in the third day of the larval life, it's already decided which larva is going to become a queen and which larva is going to be a worker. That's not true in the wasps. So the very first question that I asked is that if they all look alike, there must be some difference. There must be, the queen must be behaving differently. So how does the queen behave and how do the workers behave is the first question that I asked when I started studying this. Because it is obvious that this is an important question to ask. Now, how do you answer this question? When you do science, don't assume anything. Don't worry about what others have done. The best guide to doing science is common sense. Only when common sense fails, go to something else. We often think that common sense is too simple for science. For science, you must have something great. No, start with common sense. So I said, let me make a list of the behaviors of the wasp in plain English. So I took a notebook and a pencil and I started writing down, what do the wasps do? They sit, they fly, they move, they feed, they bite each other, they eat. So make a long list. And as you observe, my list grew to 100 items. Now I said, which of these 100 does the queen do and which of these 100 do the workers do? That might tell me how the queen behaves differently from the workers. And to my surprise, I found that the queen did all 100 and the workers did 99 because they did not lay eggs. That was the only thing they did not do, which means there was no difference between the queen and the worker, even in their behavior. But this can't be the whole story. Maybe the difference is quantitative, not qualitative. Everybody does everything, but not the same amount of everything. So I asked whether the queen may be doing things differently. So how to find this? So I decided that I'm going to measure the amount of time each wasp spends in each behavior. So I'm going to basically make a time budget. We call this a time activity budget. How much time each wasp spends in sitting, in feeding the larvae, in fighting, in biting, you measure this. So I developed some simple methods to measure these things. Then I got a large data matrix. So I had wasps and I had behaviors. So I had some 20 wasps and I had 100 behaviors. And in each cell, I had the proportion of time. Now I'm talking of daylight hours because the wasps seem to do very little at night. So during daylight, what proportion of the time does each wasp spend in each kind of behavior? Now I had a large data matrix and I needed to do some statistical analysis of this. So since the data was this kind, I decided that we need some kind of multivariate testing. So I used two methods. I used the method called principal components analysis, followed by a method called cluster analysis, basically to try to reduce the noise in this and try to understand it better. And the net result of all of this exercise was very interesting. It turned out, to our great surprise, that in every colony, we could classify wasps into three categories. Now, this is not queen versus worker. In fact, I ignored the fact that one is a queen and the rest was workers. I treated all wasps equally in my study, in my observation, in my analysis, in my computer programming, and I found there are three kinds of wasps. And based on their major characteristics, I named them as sitters, fighters, and foragers. Now, the sitters spent an overwhelming proportion of time apparently doing nothing, just being lazy, just sitting there. The fighters were these aggressive individuals, and they were constantly interacting with other individuals. So the wasps show very different kinds of aggressive behavior. One wasp may bite another wasp, it may chase another wasp, it may hold the leg or antenna of wing in the mouth and make immobilize that wasp. It may chase, it may peck the wasp. So these are all, we call these dominance subordinate. So the one who bites is dominant, the one who gets bitten is subordinate. The one who chases is dominant, the one who gets chased is subordinate. I'll talk a little bit more about these dominant subordinate behaviors. So these are the wasps which indulge much more than others in these dominance interactions, and I call them the fighters. Then there were many wasps who would go out and bring food and work for the colony, especially work outside, and I called them the foragers. So I had three groups of wasps. Now, the nice thing about this is I had not specifically decided to look at the queen or the worker separately. That's nice because now post facto I could go and ask, where is the queen? Is she a sitter? Is she a fighter? Or is she a forager? Now, common sense suggested and previous literature suggested that the queen must be a fighter. Because in such wars which had been studied by other people elsewhere, it had been shown that the queen is a very aggressive, very nasty individual. She, in fact, rules by aggression. She physically harasses her workers and makes them work. She uses aggression both to make the workers work and to prevent the workers from becoming queens, so that she has the monopoly of reproduction. So one would expect that the queens are fighters. There is nothing as valuable as a failed experiment. There's nothing as valuable as an experiment that does not give you the experiment. Exactly the opposite of practicals that we give our students. You get this answer. In our species, colony after colony, the queens were not fighters. The queens were lazy sitters. The queen in the species is a non-aggressive, even non-interactive, meek and docile city. So how does the queen behave? This is the answer. How can you have confidence in the answer? How can this be true? Because if this is true, if the queen is such a meek and docile sitter, how does she ever become a queen? Why would the workers accept her as their queen? If the queen is a nasty individual who is always bullying you, always biting you, then maybe you have no choice. But if the queen is some meek, docile sitter, she's sitting somewhere, not even interacting with you, why should you accept her authority? So you can see how a new question arises. We could not have asked this question before we answered the previous question. This question didn't exist. But now this question exists, and it lowers our confidence in the previous answer. And we say, we put that answer on hold, and we will do this. Till we answer this question, we will not say, we will not have confidence in that. This is how science progresses. This is how science should progress. All science is work in progress. So now we have to answer this question. How does she become a queen in the first place? And in order to answer this question, you have to do a different strategy. You have to design a different kind of strategy. And here we decided that we needed, see, what I showed you so far was observation of an undisturbed colony. The only disturbance we had caused was the marking groups. Now we realize that by simply observing, we are not going to be able to answer this question. We need to do an experiment. And we have to design an experiment which helps us to answer this question. And the experiment we designed is the following. We said, if we want to answer the question, how does the wasp become a queen? What should we do? We should watch at that time she is becoming a queen. How do you watch her when she's becoming queen? You remove the previous queen and one of the workers will become the queen and then you can watch them. So we designed an experiment that lasted 3 days. On the first day, we would watch the whole colony along with the queen. Of course, all the mark, we would study them for the whole day. Now, what does study mean? We make time activity budget. We determine how much time each was spent in each behavior. Early next morning, we take a forceps and quietly remove the queen very early in the morning, so there is not much disturbance. And a little later, when the workers wake up, there is no queen. So now we have a queenless colony. You spend all of the next day restudying this queenless colony. Again, restudying means reconstruct time activity, but just for all the one. How do they now spend their time in different? Now, when we did this experiment for the first time, at the end of the day, my student said, you know, whether you like it or not, whether she looks different or not, we have some respect for a queen. So when we removed the queen, we put her in a bottle, we gave her a little bit of money. So put care of her. At the end of the day, my student said, what do I do with this queen sitting in the bottle? When the experiment is over, why don't we just put her back in our colleagues and why we have no particular interest. But we found to a great surprise that when we put her back, she just went back as if nothing had happened, started being the queen. I said, let's study them again on the third day. So we recalculated the time activity budget for all the was, including the queen, on the third day. So we had a 3-day experiment, 3 sets of data, initial with the queen, without the queen, and with a reintroduction. There's nothing like getting very surprising results. Now, this experiment was very surprising. We found that this species is a reasonably peaceful society. There is some dominance about interaction, some aggression, some fighting, some chasing, but overall, I would describe it as reasonably peaceful. But when we removed the queen, all hell broke loose. From a reasonably peaceful society, it became an extremely violent society. There was an enormous amount of aggression measured in terms of these dominance behaviors, in terms of these aggressive behaviors. The number of acts of aggression measured per hour of time, in some cases doubled, quadrupled, 5 times, 10 times, 20 times. We even recorded a 30-fold increase in aggression in the colony on removal. That itself was a great surprise. But even greater surprise was that all of this aggression was shown by one single worker became hyper-aggressive. This is what the data would look like. Day 1, almost nobody is aggressive. Almost nobody is aggressive. Day 2, one individual shoots up her aggression. When we return the queen, she begins to become non-aggressive again. In fact, if we don't return the queen, she will slowly lose her aggression. She will come down to nearly 0. And this is the time at which she will become the new queen. So we call her the potential queen. So the answer is, potential queens begin their careers as very aggressive individuals, and only later they become meek and docile. So how does a queen individual become a queen? This is the answer to that. Now you can see that this answer, although we're going to put this answer on board, it increases the confidence in our previous answer that the queen is a meek and docile citizen. That now becomes a little bit more believable, not entirely, but a little bit more believable. Because there is still another serious problem. If the queen is such a meek and docile sitter, how does she prevent the workers from becoming new queens? Because in other species, it is through constant physical aggression that the queen prevents others from becoming queens. But our queen is a meek and docile sitter. She was aggressive only for about a week, first week of her life, but she may be the queen for months together. And during that period, nobody challenges her. Nobody becomes a queen. How is that possible if she is a meek? So you can see we still don't have full confidence in our answer that the queen is a meek and docile. Now we have to answer this question. To answer this question, you have to set up another kind of study, do another kind of experiment. And here what we did was we hypothesized that the queen may afford to be meek and docile and not be nasty and aggressive, because she may be producing chemical substances through which she can control the reproduction. And this is not fantasy because this is precisely what the queens of advanced insect societies do. Honeybees, ants. In an ant colony, there may be 100,000 individuals, 1 million individuals. It's impossible for the queen to go and bite each one of them. She produces chemical substance, and this chemical substance actually prevents the workers from developing their. So even though this is a primitively used social insect, maybe the wasp hasn't read the books, she doesn't know that she's primitively used social, she's actually started producing pheromone. Maybe that is possible. If that is true, is this pheromone a volatile substance or a non-volatile substance? So we designed a different kind of experiment. We brought a colony to the lab, we put them in a cage, and we did the following kind of experiment. On the first day, inside the cage, we studied all the wasps. And basically studied means how they distribute their time. On day 2, we removed all the wasps and we took a blade and we cut the nest roughly equally in 2 parts, roughly equal amount of brood on both sides, and we inserted a wire mesh between these 2. Now the function of the wire mesh was to prevent wasps from going through, but allow any volatiles, prevent non-volatiles from going through, and wasps from going through, but allow volatiles. Once we fixed this wire mesh in place, we had 2 halves of the nest. At present, yet no wasps. Then we picked up 1 wasp at a time, tossed a coin, and decided heads you go left, tails you go right. Next wasp, heads you go left, tails you go right. And finally, we picked up the queen. You're supposed to treat the queen like everybody else. You also heads you go left, tails you go right. So at the end, we had 1 half with a randomly chosen half of the workers and the queen, the other half with the remaining workers without the queen. Now we studied the wasps on both sides. We studied the entire cage once again on day 2. On day 3, the equivalent of returning the queen, we picked the queen from her side and moved her to the opposite side without disturbing the workers and re-studied it on the 3rd day. What would we expect to happen? We had clear-cut predictions. If the queen pheromone is volatile, both nest fragments should behave as if they have a queen, because the workers on the other side can smell the queen, they have a queen, so nothing should happen. But if the queen substance is non-volatile, then of course, the queenless side should think they don't have a queen anymore, because the non-volatile is not coming through, and there should be a hyper-aggressive potential queen, and they should behave like a different colony and get their own queen. This particular kind of experiment, which is very tedious to do because you have to cut the mesh, put the mesh, cut the nest, and introduce, but it's a very, very powerful experimental paradigm. And we have used this particular experimental design to answer many other questions. I'll have at least one more occasion to tell you about this. So when I made this slide, this particular experiment had been done 24 times. And in all 24 cases, prediction 2 was upheld. That means on the queenless side, one individual became hyper-aggressive. And in fact, what happened was that here there was a hyper-aggressive individual. When the queen was moved to this side, this hyper-aggressive individual ceased to be aggressive, but a new individual here became hyper-aggressive, because now that is the queenless side. This is what the data looks like. Day 1, no, there's no aggression. Day 2, one individual becomes hyper-aggressive. We call her the potential queen 1 on the queenless side. When we moved the queen to her side, she dropped her aggression significantly. And another individual who was showing no aggression on day 2 or day 1 became hyper-aggressive. It's interesting to think that these numbers, we could only get this post facto. Because on day 1, we didn't know who the potential queen 1 was or who the potential queen 2. We only identified the post facto. That today you are aggressive, what did you do yesterday? Today you are aggressive, what did you do the day before yesterday? Because day before yesterday, we didn't know who you are. So we got this post facto, and you can see very clear-cut pattern supporting prediction 2. In fact, the new queenless side now has a potential queen, and the previous day's potential queen goes back to her. So the answer is, Propylidia marginata queens appear to use non-volatile pheromones to inhibit worker reproduction, at least to advertise themselves to the So you can begin to see, now we have a little bit more confidence in our conclusion that the queen of our species is a meek and docile sitter, because we know how she manages to do it. So as we go on, the more questions we answer down the line, the confidence in the previous questions gradually starts increasing. So it's a very gradual process. So if you ask me about my confidence in the answers we have got in the last 1 year, very low, because we have to go further to get confidence. Now, another question arises. If this is a non-volatile, how do the workers perceive the queen? How do they know that they have a queen? Now, I won't go into the details because it took us 2 years to answer this question. We tried all kinds of things. We failed most of the time. But finally, we came to the conclusion that the queen, without going into the experimental details, because I want to show you more questions and answers in the time that I have. So I won't go into details, but our answer was the queen actually applies her pheromone to the nest surface. So as she walks on the nest, she's actually applying this non-venomous substance to the nest substance, and it is from there that the workers are actually picking it up. Then the question arises, okay, if that's what's happening, what is the source of this queen? Now we found that the queen was rubbing the underside of her abdomen. So something coming out from the underside, you turn around, and in fact there's a hole there. There's an opening, and there is a duct which leads to a gland called the divorce gland. So this is called the divorce gland. So we suspected that this is the source, or at least one of the sources of the divorce gland. So we took that gland out, we crushed it, we mashed it, made a crude extract. And we said, let's ask, in fact, if this is where the pheromone is. And we had to do a bioassay. And our bioassay is very simple because we know that in the absence of the queen, the potential queen is hyperaggressive. When you return the queen, she drops her aggression. So now I remove a queen, I wait for the potential queen to become hyperaggressive. Instead of returning the queen, who will then go and apply the pheromone, I will apply the pheromone and see if I can make the potential queen lose her aggression. Can I pretend to be the queen? So the bioassay worked like this. What you see in this panel is, this is session 1. Queen is present, no aggression. Remove the queen, huge amount of aggression. Apply the pheromone, significant reduction in the aggression. Not as much as by the queen, because I'm not as good as the queen. My student is not as good as the queen, but there is a statistically significant reduction in aggression when you apply the substance. Control experiment, very important. Apply the same divorce gland you extract from a worker. That doesn't work. So worker divorce gland doesn't have substance, nor does applying the ringer work. So you've taken care of disturbance, you've taken care of worker gland. So it looks like this is the non-volatile substance. Now, I should tell you that I am very proud that we should do low-cost research. We should be inexpensive. And my students sometimes tease me about this. By the time we reach this stage, they said, now you have to buy a GC MS machine. Now you can't pretend that you would. And no. First we do a bioassay. After you do a bioassay, maybe, but not before a bioassay. But once the bioassay was done, we went to a colleague and said, can you please run this on your GC? Because we do low-cost research. And they ran it on the GC. And we found that the queen and worker divorce glands contain a series of linear monomethyl and dimethyl branch alkanes with 21 to 33 carbons. Considering all individuals, there are about 30 different compounds. No compound or set of compounds are present exclusively in the queen of the worker. So what work is a cocktail? And in the cocktail, there are, as you can see here, there are different proportions. These are the areas under the curve for each of the compounds. And it is this unique blend that makes you a queen or a worker. Otherwise, all the compounds. And this is actually not so surprising. Anybody who can smell 2 different whiskeys and say, oh, this is different from this, will know that it's a blend and you can distinguish blend. And workers obviously are able to distinguish blend. And the other proof we had was we asked our computer to classify an individual either as a queen or as a worker
based on data. What is the data? The area under the curve for each of these peaks. So we gave this to the computer program, and the computer program with 100% accurately classified all queens as queens and all workers as workers. So we have some confidence that this is how the system works. We suspect that the source of the queen pheromone is in fact a divorce gland. We cannot say this compound, but it is this cocktail of the queen, which is different from the worker cocktail, which really makes the worker say, this is my queen. Now, the next question is not one that directly arose from this, but it arose from all the previous. In fact, whenever I gave a talk, people asked me, you remove the queen and one worker becomes hyperactive and becomes the next queen. Who is this lucky individual? Who is the next individual? Can you predict this identity of the next queen in the presence of? As humans, we are very interested in predicting who the next prime minister will be, who the next director will be, who the next chief would be. Very interested. So everybody said, who is the next? And I would say, doesn't look like it I can predict, because we don't see anything different. So for every time I would say, it doesn't look like. But these questions became so persistent that I had to do something. I had to do some real research to see if we can predict this individual. Otherwise, I was hand waving and saying no. And so at one point, I decided that I'm going to investigate. I'm going to solve this problem. I'm going to try and see if we can predict. By then, I was already a professor. When a professor decides that he has to do something, what do? Next PhD student is given the job. So the next PhD student who came to my lab, I said, you have to predict the identity of the individual who will become the next queen, you get your PhD. So this student actually worked very hard, spent 5 years, did many experiments, discovered many things, published many papers, but she threw up her hands and said, I cannot predict. To give you just one example of the kind of things she did, she showed that the potential queens are mixed up with the worker, and you cannot tell. In fact, if at all the potential queen is more worker-like, queen-like, but there are many workers. So the answer at that time was, no, we cannot predict the identity of the potential in the presence of the queen. The potential queen is not unique by any criterion. She is not the most dominant individual in terms of behavior. She doesn't have any unique behavior. She is not even having the highest amount of low ovarian development. See, the queen has a lot of ovarian development. The workers have some residual, and she is not the best among those. She is not unique in any way, so we cannot identify. At this stage, I had already studying this species for a very long time, and I had developed a kind of love. We can't help loving the previous. I secretly was very proud that we failed. This war still has some secrets which we don't have. We cannot tell. However, there was one student. I'll tell you about next 2-3 students. So at this stage, I said, no, we cannot predict. And that's sort of, I'm not very upset about it. It seems like a very nice thing because this wasp is really very clever. One student came to my lab and said, okay, we cannot predict who the queen is. Do the wasps know who their next queen is before the queen dies? Many people said, this is not a scientific thing. How would you know what the wasps know? How would you know the mind of the wasp? But this student was very persistent. So we spent a lot of time thinking about this. Can we really perform an experiment which will allow us to answer this question? Do the wasps know who their next queen is? Believe me, so many people put cold water on our thing because everybody said that. But we spent out. And one day we came up with an experiment. We did the experiment and we found that the experiment suggested that the wasps know. After 2 nights, sleepless nights, we decided that this experiment is not a good. It's a flawed experiment. There are other possible explanations. Went back to the drawing board, spent more time, and we came up with another experiment design, and I think that worked. And I will explain to you what that experiment is, how we went about answering this question. So is it possible? So first of all, I said, let's make this question a little bit more scientific. Not saying do the wasps know. So we said, let's make it sound scientific. Is it possible that there's a hair designate? Even though we cannot identify her, do the other wasps somehow know who this hair designate? Now, I told you about this experiment where we cut the nest in half and put a wire mesh. That design came very useful here, and we decided to test this hypothesis. Now, change of strategy. Here, the philosophy was completely different. Here we said, we'll do science in a different mode. We are going to assume that there is a hair designate. We are going to assume that all the wasps know who the hair designate. Now we do an experiment and we predict how the wasp should behave in our experiment. If they behave in the way we predict, then our assumptions are correct. If they don't, our assumptions are. So we make the assumption beforehand. And let me explain, this is a very tedious experiment, but very easy to understand. Let me explain this through a little animation. So imagine that this is the nest. We have cut it in half with a wire mesh. Queen and half the workers on one side, the remaining workers on the other side. Now, if you do such an experiment, and if our assumptions are correct, there is a hair designate, and everybody knows who that is, except us. All the wasps know who that hair designate. What would we expect? We would expect that if the hair designate is on the opposite side, as it could happen just by chance alone, we already know that these wasps think they don't have a queen because the substance is non-volatile. So the hair designate should immediately become hyperaggressive, and she should become a potential queen on her side, as we might say. But she is here by accident. She is supposed to be the true hair designate for the entire colony, not just for her side. She should also be acceptable as next queen for these workers. How do you test that? Interchange the potential queen and the queen. Don't disturb the workers. Take the potential queen from one side. You can recognize that she's hyper-aggressive. Other side, move the queen on this side. And she should be a queen individual, move her back. There should be no problem. She should be acceptable both. However, this can only happen by chance in about half the experiment. In the other half experiment, the unfortunate hair designate sitting next to the queen, and there's nothing she can do about it because there is a queen. Nobody will accept her if she becomes hyperaggressive. What would you expect to happen? In this, you would expect that the best amongst those workers would become hyperaggressive. So let's call her potential. She'll become the potential queen because she's the best individual. But she is only on this side. So what do? We bring her face to face. We move her to the other side and we bring her to this side, face to face with the hair designate. Now we expected that all hell should break loose because the real hair designate is sitting here and an individual has become hyper-aggressive has come. And in fact, we predicted that this should lose and this should become potential queen. So in the sequence, she is number 2, so we call her 2. But that's not the end of the story. The acid test for this individual is when I move her to this side, she should be acceptable to that side because she is the true hair designate for the whole colony and she should be acceptable to both sides. So we have 2 predictions. The first individual to become hyperaggressive, PQ1, should be unacceptable to the opposite side in about half the experiment. And in a second individual should emerge as the potential. But the potential queen number 2 should be accepted to both sides. There should never be a PQ3. There should never be a third individual. There should never be 3 contenders to the throne if you cut the nest in 2 parts. These are the predictions. Now, these experiments, as you can imagine, are extremely tedious to do. But my student was all fired up, 2 years of planning. So she did this experiment over the next 2 years, 8 times. Now, she could have done more, but in some of the experiments, the wasps just get tired and they give up because it's just too much disturbance. But she did this experiment 8 times. In three of the 8 experiments, there was no PQ2, and the first one was successful on both sides. But in the remaining 5 experiments, the first one had to lose her aggression, but the second one was successful on both sides. So in fact, we can say that the PQ2 emerged in about 50% of the cases, which is statistically significant, No PQ3 ever emerged. So both predictions were upheld, suggesting that there is a cryptic hair designate. So there is a hair designate. We call her cryptic. She's cryptic to us. Probably not to the wasp. In fact, not to the wasp. Now, this was a very interesting situation because every time people ask me, do you know who the potential queen is? I would sort of say, no. Now we could say, we don't know, but we know that the wasps know. In fact, we published a paper with the title, We Know That the Wasps Know. And I'm very grateful to the referees and editors journal who allowed me to keep this title. So now we could probably say, we don't know, but we know that the wasps. Now, why do we have confidence that the wasps know? It is remarkable that the potential queen number 2 was never challenged. She did not receive a single act of aggression by the potential queen 1 or by any worker, either when she arose or when she was challenged. And that is why we think the hair designate was known to everybody. Everybody knew, including the potential queen 1 knew that it's not my turn. And that is why we think that the wasps know who their potential is. Now, the next student, see, I am very satisfied with this, but students are not satisfied. Next student said, and of course, junior students love to challenge senior students. So the next student came and said, okay, you've done all of this. You are saying there is a hair designate. How do you know there is only one hair designate? Maybe there are many hair designates standing in a queue. Do the experiment. And what this student did, it's a clever experiment. He identified a colony, identified the queen, removed the queen, and identified the potential. Now, as soon as he identified the queen, potential queen by her hyperaggression, he removed her to see what would happen next. So there is a hair designate, even though we cannot identify her. Is there just one hair designate, or is there a queue of hair designates patiently awaiting their turn? So the experiment results were spectacular. In the presence of the queen, no aggression whatsoever. Remove the queen, one and only one individual becomes hyperaggressive. Nobody else becomes hyperaggressive. Remove that individual, of the rest, one and only one individual becomes hyperaggressive. Remove her, one. At the end of 5 such experiments, he said, I have proved the principle. And these individuals were unique because they were not challenged by anybody. In fact, I'll blow up the data and show you these blue bars, which are essentially 0, record the number of acts of aggression that the potential queens received, which were essentially 0. So only one individual will become hyper-aggressive, beat up everybody else. Nobody would beat her back. And that is why we think that this system works. There is a serial. So there is a long reproductive queue of at least 5 potential queens, but perhaps it's even longer. Maybe all individuals are actually standing in the queue waiting for their turn. For the next student, this became too much. That we know that the wasps know there is not one, but there are many, and still we admit that we cannot identify ourselves. So she said, I'm going to my PhD on, I'm going to come back and do my PhD trying to break the potential. Now this time I actually dissuaded her. I said, this is a very tough problem. It's not a PhD problem. It's a lifetime career. So you do your PhD on something else. And she kept this in her mind. She did her PhD on something else. She went abroad and she found out that her supervisor, her postdoc supervisor, had a technique called multi-layer network analysis. And she wrote to me excitedly saying, maybe if I use that technique, we will be able to identify the potential queen. I said, go ahead. We worked on that. And in fact, she was able to use this. In the past, each social situation was examined in isolation. We have now, this new student, after she went abroad, used the novel analytic approach of multi-layer network. This combines multiple social situations and produces a single mathematical object. A multi-layer network accounts for the differential impacts of different social situations on each other as well as on the global dynamics. And a multi-layer network measures a quantity, a mathematical quantity, which is called versatility. And this applies for all situations. So basically the question we ask, is the potential queen the most versatile individual in the colony before we remove the queen? So these are the different kinds of situations: aggressive situation, food exchange situation, liquid food exchange, solid food exchange, and spatial overlap. When you take each one of these, you cannot identify the potential. She's not unique. But when you combine all of these, it turns out that actually you can show that potential queen is unique compared to all other individuals. So in fact, at this stage, we come to the conclusion that we can probably identify the potential queen after all. Why do I say probably? Because work in progress. Put this answer on hold. What would I like to do? What yet been done is I would like to take a colony, collect data, run multivariate net analysis, have in my hand my prediction. Who is the potential? Then go and remove the queen and see if my prediction. We haven't done that. Nobody done it. That can be done. But there is yet another puzzling new question. We use multivariate network analysis. What do the WASPs do? As far as we know, they don't have access to multivariate network analysis. How do they identify their next queen? We have no idea. Maybe future work will tell. Now I want to switch gears and say, it's so wonderful, this species. There is so much cooperation, so much order. It's so beautifully done. Everybody knows who the next queen is, and all the potential queens are lined up in queue. They don't go out of turn. All of this is true. Where is the war? And where is the conflict? In fact, the reason why I put this in the end, and the reason why I've left very little time for this, is because indeed, throughout my research career, I've been waiting to see the conflict. I'm waiting to see the fight. The WASPs are so well behaved, so cooperative, that you actually won't see these things. And we somehow wanted to see. We thought that when we bring the hair designate and the potential queen one face to face, there will be a fight. There wasn't a fight. The earlier potential queen voluntarily went back to working. So we wanted to see a fight. So we have now created some situations where we can see conflict. One question we asked is, how does the queen maintain her status? And why does she sometimes lose it? Because in nature, from time to time, the queen is replaced by another. So this happens in nature. Why does she maintain sometimes? Why does she lose it? So we decided to do an experiment that we can kill two birds in one shot. We can answer this question, how does she maintain her status? Why does she lose it? Also provide more evidence for our conclusion that the pheromone is non-volatile. So we decided to prevent the queen from applying her pheromone. If we prevent the queen from applying her pheromone, she should lose her status. She should be challenged, somebody else should. Now, how do you prevent the queen from applying? We know it's produced in Dufort-Gly. We know that there's a duct. We know that it comes down to the underside of the apple. So people suggested that we close this open. In fact, people said you put wax. Somebody said you put nail polish. Now, all of this is too disturbing to walk. I like to disturb the wall and so I said none of this. We designed a very different experiment. What we did was that we went to nature, studied a colony in nature, identified the queen. Then we collected all the wasps. The queen and all the workers were transferred into a cage, but no nest. We didn't give them a nest. Question is, now what will the queen do? Where will she apply? She won't be able to apply her pheromone, and therefore she should be overthrown. But she was not overthrown. Why was she not overthrown? The queen continued to rub her abdomen quite frantically on the walls of the cage, all over. She was literally like a mad person applying all over the cage. That is why she was not overthrown. But if she applies the same quantity of pheromone to the vast surface of the cage, rather than the small nest, it should be very, very dilute. And there should be some effect of this dilution. We must see some effect of this dilution. You must always ask these questions which try to disprove your own answer. Then you are successful. We said that we don't believe this because it can't be true. We can't say she's applying on the cage and therefore she's not overthrown. It can't be, because what she's applying on a 2 centimeter by 2 centimeter nest, now she's applying on a 15 cube inch. So there must be some signature of this dilution. Otherwise, you cannot trust your answer. And we looked for signature and we found it. In fact, the workers attacked her. And they never do this in natural conflict. So we saw a fight. We saw the fight between the workers and the queen because it was very, very tight. Even more interesting, for the first ever time, we found that the queen retired. The meek and docile sitter who sits in a corner, doesn't interact with anybody, fought back. So when necessary, she is able to fight back. And so we answered the question, how does she maintain her status? By producing good pheromone, good quality, sufficient pheromone. Why does she lose it sometimes? Because she doesn't have enough pheromone. But we also discovered in the process that workers can attack the queen if necessary. Even more interesting, the queen can fight back if necessary. So she may be a meek and docile sitter, but she hasn't lost the ability to. So there is potential for conflict, but it is prevented. That is what is nice. There is potential for conflict. Workers can attack the queen. Queen can fight back, but they behave in such a civilized manner. They control all of this and they maintain. That is what I mean by balancing cooperation conflict. We asked another question. We thought there's another way to see even more conflict. What about conflict with outsiders? We expect more conflict when there are outsiders. How do they treat wasps from other colonies of the same species? So here we did the following experiment. When the members of one colony were introduced into the cage of so we had two cages, all wasps, not all wasps studied. We took all the wasps from one cage without the nest and brought them to the other cage. So now we had this cage with a nest and its resident wasp and alien wasp who did not have this. So the alien was in foreign territory, no nest, and this. So now we wanted to see, and we found the result was very interesting. The resident wasp had three kinds of reaction to the aliens. The young alien wasps were admitted freely into the colony. No problem, come and join my colony. Old alien workers were allowed to live, but only away from the nest. You can live in that corner of the cage, but don't come anywhere near the nest. The alien queen was located and physically torn to pieces. So the resident showed a very nuanced differential treatment. So we saw fight, we saw conflict, but we also discovered the ability of the wasp to show this very nuanced kind. Now, given this, the next obvious question was, these young aliens are accepted. What is their fate? What becomes of them? So we did a very detailed experiment where we deliberately brought aliens. And in fact, we brought them from very far away because we didn't want them to be genetically related. So we went to Mysore and collected some wasps and we took the young ones from those and introduced it to Bangalore colonies. And we found that young alien workers who are accepted into foreign colonies become completely integrated into their foster colonies and become normal workers and may even become future queens of that colony. In fact, the probability of a foreign worker becoming a queen, we showed, is statistically directly proportional to the number of foreigners. So there's absolutely no discrimination. Once they enter, they become card-carrying members of the colony. So there is war and there is peace. The wasps have a great propensity to make war with outsiders and an equally great propensity to maintain peace with insiders. From an evolutionary point of view, war with outsiders is easy to explain. And peace with insiders is less easy to explain. Why? The wasps are not clones of each other. They still have genetic conflict. That's hard to explain. But war with outsiders is not of much use unless one can combine it with peace with insiders. If you have war with outsiders and war inside, nothing is going to be gained. It is this dual strategy, this ability to tread the fine balance between conflict and cooperation, we think accounts for the success of insect society. And insect societies are hugely successful. In fact, we call them superorganisms. That's why we think they are so successful. I want to end by saying that I have been writing about these things for various kinds of audiences. My first book is called Survival Strategies, and this explains our field in a very simple manner and a little bit of our research. Some years later, I published a monograph on about 20 years of research on this wasp. More recently, this was referred to in the introduction. I published a book called Experiments in Animal Behavior. In fact, the subtitle is probably more important, Cutting Edge Research at Trifling Cost. In this book, there are 16 chapters. The 15 chapters are take examples of research that you can do with almost no money. And the last chapter talks more generally about the importance of cutting edge research, why it is so important to do, but why is it so difficult to do. Not because you cannot do it, but because there is no prestige in doing it. And how we can solve that problem and create prestige. So this book is freely available on the internet. I said to my publisher, I cannot write a book called Cutting Edge Research at Trifling Cost and make the book costly. So the book is actually completely free. You can see there's a website here. You can access this book freely. I also think it's very important for scientists to communicate with the public. Again, as I said, not just the products, but the process of science. So I regularly write a column in the Wire Science called More Fun Than Fun. And my goal here is to transmit the joy, spirit and the joy of science to a wide article. I have written 40 articles so far. Again, these are all freely available at this site. Just to give you a sample of the kinds of articles I've written. This is called Was Food Be Queen? This is called Caucus. What can Caucus teach us about parental care? The delicate truths between cleaner fish and their clients. What do dogs and other animals do all day and night? The exquisite wax palace of honeybees. Chasing wasps on a bicycle and getting a PhD. There are 40 such articles. Again, those who are interested can look at the board, freely available, and I promise to write more and they will be uploaded into this site. I want to close by again saying the fun of this research for me was because of my students. In fact, as you have seen, I didn't need any sophisticated, there was no sophisticated instrumentation. There was nothing that I needed. In fact, in India, it's very fashionable to have something in places like RCB, every lab is a. But in many small institutions, there's one room which is called a sophisticated instrument facility. And that is the people are very proud of it. So this is from the to sophisticated instruments. I have often been taken in university to say, oh, come and see our sophisticated instrument facility. Just show me lots of that. And people come to our lab and say, where is your sophisticated instrument? You have one. Yes, I do. These are my sophisticated. I greatly enjoy working with students, arguing with students, debating with students, much more than fighting with instruments. And that gives me great fun. Yes, I have been funded by DSC, SCRB, Ministry of Environment for SCSR, DBT, but 90% of the money I've used to pay students. So that's what I really mean. And that we are doing science is great fun. I'm very glad to have had the opportunity to talk to you. Wish you a wonderful RCB day. Thank you.
Thank you, Professor Gadagkar, for very, very, fascinating journey. And finally, you put everybody in the beehive, right? I was, while he was talking, he was also citing his publications. And I wanted to draw your attention when he says that you can do good science with very little money or no money. And there are many, journals that he is published in, which we don't know about, but there was certainly one journal, PNAS. And when we think of PNAS, we think that we really need to have sophisticated instrumentation to do experiments to publish something in there. But it is the idea and the solution and the strategy that one uses to answer a certain questions. And that is the example. That is one of the example I think we can draw from his work. So thank you very much. I wonder if there are any questions and whether you would be willing to take some of the questions. So I'm sure there are many questions, but let's take 6.
Usually questions give me ideas to the next experiment.
No, even I have many questions, but I will ask you later. But let us take 6 questions from students. And yes, Kashpatika, you go ahead.
That is a great talk. I have one question. My question is, can the developing aggression potentially be the absence of the gene actually be because of the change in their pocket in their reports? Because I mentioned saying the queen has a very different cocktail, the same chemical compound and no. So there should be somewhere that it change in the worker that becomes the protected we have. So can that be the reason for their attention?
No, it's but that's not the reason. It is a problem. So, unfortunately, getting this cocktail is destructive, right? So we took one colony, we removed the queen. In fact, before removing the queen, we removed the queen as soon as injected the hydrogen connected. In another colony, we removed the queen, waited for 24 hours, removed the aggressive. Third colony waited for 36 hours and found. So we did this serial experiment and we plotted it. So what we showed was matter of 1 week, the potential queens drop their aggression from very high as soon as you remove the queen to essentially zero. They develop their ovaries from worker-like ovaries to queen-like ovaries, and their cocktail plotted in multi-dimensional space, there are 30 compounds. It moves from a cocktail that is indistinguishable from that of a worker to one that is indistinguishable from that of a queen. 5 to 7. So the aggression, we see the same. But within about 5 to 7 days, the potential phase cocktail becomes indistinguishable.
What could be the reason for their sudden development of depression? Apart from the fact that the queen is removed.
Because, okay, so then you have to know. And this is something we investigate again. So our obvious hypothesis was, I don't get how queen is going to want. So now let me pick up everybody and be an aggressive queen like the other 50 until I get mine. So first, we showed aggression to the. So we said we made prediction. What would be our prediction? If the function of this hyper-aggression of the potential is in order to suffer. He said that she should show more aggression if there are more workers. She should show more aggression toward more. She should show more aggression toward workers who are better than me. So we were lost. What is when we put on the table, it's extremely unlikely, a very counterintuitive. And I don't know to. If we can prove counterintuitive record, so we deliver it, but we also have to make a very counterintuitive prediction. So our counterintuitive hypothesis was the function of the hyperaggression protein is not to suppress the worker, but she needs it for her own good. Is that good? Fact, aggression. Now, so reserves. Hormone making aggression. So think of exercise. When you exercise, you change the result. So she may actually be crazy. But when the crazy prediction is, if I am very few workers, see what would be if a lot of workers actually so it will take me longer to develop. But our prediction is opposite. If you have more workers, you can actually take steps. This created a colony in which there are potential with nobody to show. It took a long time to develop. When she had individuals, she had to spend 5 minutes, she developed her. So we actually published a paper with the title Possible Novel Functional Aggression. Because for aggression itself, we believe we have suggested a novel. And we have argued that people who study aggression should pay more attention to the aggressor, not just the victim. Everybody is looking at the victim. Whatever the aggressor, whatever the physiological. So we think we have suggested a new. Thank you for that.
Yeah, go ahead.
That was a good answer, and I took it very. I just the question just came up in my mind after what she asked. Is it possible that the water, the groundwater that is on the case or into the nest, the reduction in this substance, when it creates, that makes the potential weak because of some external stimuli that this body has.
That would be fine. But interestingly, what we have, so there is something that everybody knows that this is the next contact. And she is waiting for that cue to speak English. If there is no queen, I have to become a queen. So basically, the workers are detecting the presence of actual queen by this.
Right, go ahead.
Yes.
Now one over there.
Hello, sir, this is a very nice one. Sir, you said the pheromones has also inhibit the reproductive activity of the other workers. So, is there a potential queen pheromones with the same ability as the queen has to inhibit the reproductive efficiency of the other workers? So, is that much affected in the potential queen, the pheromones which is found? Is that much potential to inhibit the reproducibility of other workers?
Only after me, not before me.
So when it is the potential queen, so the pheromones is exactly same as worker or
When you have queen, everybody else comes. She is not a calling queen pheromone. She is not able to lay because of the potential. About a week later, she has developed the queen pheromone, reduced her aggression, developed the body, name as first take, then we call her the queen. So the label potential is for that 1 week period when you have become hyperactive, but you have not yet laid yourself. As soon as she gets the first thing, we call her. So once she becomes queen, then she has enough for a long rest. But during this period, she probably doesn't. But we do not think that we do. But that is why it is important that the workers know who it is. And it is this knowledge that prevents them.
Yeah, over there. Yeah, Sunny.
Wonderful talk, sir. It's feel like I'm watching probably. So, my case is when you enlist the property, the other property, so we will need the position of the client into this.
Yeah.
Because there is multi networks over there.
So now I can, that's actually one question I can tell all the things that I did tell probably have time. This student who said, I am not, I am also going to my PhD and I am going to protect it. I persuaded her not to do that. So what did she do? She studied the facial positions of the wasp. And she showed that wasps have their favorite way. They don't do this kind of thing. She and she studied this non-random spatial distribution of wasps. That was a PhD. But when she went abroad and she decided to reopen this question, in addition to all the other data that all my students had, she also had data on the spatial position. She was able to add that and as an additional social security. Then combine all of this and upward. That spatial data. Definitely have to.
And the second thing is, if you can answer it,
Yes.
the network you found in which you are specifying digital green and the PT. So all the other the lineage come out of this second, third, and fourth.
Yes.
So are they surrounding the PT in that network? Because if it's a positional property, then the DPT surrounded by the hollow trees.
Can you count my life? They are not all. Because this is very different. We have just done with money.
Now the camera can capture everything.
Can we answer these question, your previous answer? We are not here to say we know everything. We are here to say we are curious. I would be very sorry if all questions are answered. But nothing more for us to do. You can see how these. Every time I give a talk, I get questions which
go back and say, now we can do it. But this is something we have no idea.
Okay, I think in the interest of time, there are many questions, but Professor Gadagkar is going to be around. So while you are enjoying your high tea, please interact with him and get first-hand answer from him, person to person. So thank you very much, Professor Gadagkar, for this very fascinating and interesting talk. And for you, so that you remember us.
Yes.
Thank you very much.
Actually, plan.
Yes.
We will carry this to Bangalore, but
Yeah.
it should be possible. We will find a bag. for
Yeah,
you.
wonderful.
Thank you very much.
Very nice. Thank you. I think over to you people, whatever is that. Yeah.
Now we will start with the prize distribution ceremony. So I request Professor Vrati and Professor Gadagkar to present the award.
Oh, okay.
Ah.
So actually we had these presentations in the morning
Sure.
and posters
I
yesterday.
listened to some of
Yeah.
them.
So, yes, so please go ahead.
So, good evening, everyone. As you would know that in last 2 days, today and yesterday, we had lot of interesting scientific talks, ranging from pitch talks, poster presentations, and oral talks today. Just so that everyone can, appreciate, the judges had very difficult time to select top 3 or top 4 in each category. So I think a round of applause should be first of all for all the participants. So yesterday we had 22 pitch talks from students, and judges have selected top 3, and I'm calling the names in random order. They are all top 3. First name, Pooja Ghosh from Dr. Dipti Jain's lab. Second name is Gulshan Thakur from Dr. Divya's lab. Third name is Manya Batra from Dr. Shivram's lab. Someone from Dr.
Pooja Ghosh.
Shivram's lab.
It's an honor, sir, to take it.
Gulshan.
She is not around. So
Ah,
I'm from Dr. Shivram.
so you've come to collect.
Yeah, sure.
You keep the money.
Okay, followed by pitch talks, we had poster presentations from 35 students, and judges have again selected 5 best posters, not in order of merit, but all of them are categorized as top 5. First name is Simran Chabra from Dr. Manjula Kavya's lab. Second name is Nutan Sharma from our lab, Rajendra Mumtiani. Third, Poonam Yadav from Dr. Avinash Bajaj's lab. Then we have Joydeep Sharma from Dr. Sam Nephi's lab. And we have Momita Ghosh from Dr. Dipti Jain's lab.
Poonam here, congratulations. Who's next? Jaydeep. Momita.
So finally, today we had oral presentations from senior students, year 4th and 5th, and judges have selected top 4 presenters. First name is Akshata Davane from Ramu Vindranath lab.
Second name is Lavi Rastogi from Dr. Prashant Pawar's lab. Third is Vinay Sharma from Dr. Vengadathan's lab. Fourth is Shikha Raghav from Dr. Bittijan's lab. All the participants can collect their participation certificates from academic section in couple of days. With that, the prize distribution ceremony completed. Thank you. On behalf of RCB Foundation Day and RCB Science Day Organizing Committee, I take this opportunity to propose vote of thanks. Although it appears as a small two-day activity, lots of efforts are put in by several individuals in thoughtful planning, meticulous preparations, and flawless execution. It is therefore important to acknowledge the hard work and time invested in organizing this successful event. First of all, we would like to thank Executive Director, RCB Professor Sudhanshu Vrati for his guidance, support, and leadership that helped us immensely in organizing both the events successfully. Next, we would like to thank Dean RCB Professor R.P. Roy for his continuous guidance and comprehensive discussions that we had over the last couple of weeks in planning and execution of this event. We would like to appreciate the support that we got from Registrar RCB Professor Prasenjit Guchet. No institutional activity can be successfully organized without support from the administration and finance departments. We sincerely thank Dr. Sudeep Bhar, COA, RCB, and Mr. Sanjeev Goyal, FO, RCB, for their constant support. We would also like to thank our institutional IT team led by Mr. Naveen Kumar for all the IT support that they have provided in last 2 days. The quality of food and the variety of delicacies served is one of the important measure of success of an event. And we would have to thank Mr. Ramesh from ED Secretariat for taking care of all the food arrangement. We also thank him for help with other logistics. We would like to thank academic section, especially Mr. Priyanshu and Mr. Chair for their help. Most importantly, no scientific event is possible without enthusiastic and energetic participants. We would like to thank all the participants of pitch talk, poster talk, and oral presentation. And as I said, the quality of presentations can be appreciated from the fact that judges were finding it very difficult to find top 3. And in one of the sections, we actually increased the number of prizes to 4. Students would never be able to give their best without guidance and encouragement from their PIs. Therefore, we would like to thank all the faculty members of RCB for encouraging their students to participate in different types of presentations. We would like to thank the judges who spent their time in evaluating all these different types of presentation. For smooth execution of all the planned activities, a sincere and dynamic pool of student volunteers is essential. And these volunteers also played a very important role in successful organization of these 2 events. This year, Harsh Thakur, Sukirti, Mukesh, Sharon, Aparna, Shivani, Suman, Kunal, and Kriti Ahuja volunteered for 2 days. Last but not the least, I would like to thank my colleagues and I would say veteran members of this RCB Science Day and Foundation Day Committee, Organizing Committee, Dr. Dipti Jain and Dr. Divya Chandran, for their arduous efforts and skillful organization skills. which played a critical role in successful organization of these events. With this, I thank you all and invite you all to join us for high tea. Thank you.
I request all of you to please assemble back by 6 p.m. for the cultural event.